who found out about us on Native Plant Day? Okay, so I'm going to start out with some thank yous. I want to thank our Vice President, soon to be President, Karina, if you vote her in. And her husband Sergio, who's learning right now. They brought in the coffee and refreshments, but you know, it's catching, Karina. I see more people bringing in refreshments, so it's really quite the smorgasbord. And Sebastian, what did you bring for us tonight? Fruity Pebble Rice Krispies. Good grief. All right. There is, I know that we don't use cash anymore, but there is a jar for a tip. We do have a donate button. You can donate like $2 if you want on our website. It's right up on the top right hand. It's not obligatory, but it does help pay for the refreshments. I also want to thank the plant raffle donors. We have an excellent selection I see here. Of course, there's always a great group of plants from our online store folks brought by Raul Moas tonight, who's our primary grower. And he grows the cool stuff. So you can buy raffle tickets. Who is selling raffle tickets tonight? Susan in the back will sell you raffle tickets. They are 2 for $1 or 12 for $5 is my memory. So most people get a dozen. By the way, if you brought in raffle tickets or if you brought in refreshments, you can get, or if you brought in plants and you brought in refreshments, you can get raffle tickets for the cost of your refreshments. When you bring in, if you want to bring in raffle plants, please label them as best you can, or bring a blank label and we'll figure it out for you. I will say I'm a harsh judge. So if you bring in something that looks pitiful, you know, we're not the pound. Or maybe we are, maybe I'm too harsh. I will state something. So all the money brought in from the raffle ticket sales are donations. So it's not a real raffle, it's a drawing. So, please also note that we have t-shirts and mugs in the back for sale. You know, spring is here. You should wear a flowery t-shirt. It's in vogue right now. And you'll also be advertising for the society. Yes, Susan. Yes, those are very popular. Sergio is showing one now. If you go to Trader Joe's with that, I mean, come on. Whatever they're bringing their stupid tote bags, that's local art. It's not made in China. All right. Are there still free handouts and magazines somewhere? Not tonight. Sometimes people bring in old palmettos and stuff. I know she's not here, but I'm going to thank Daniela anyway. She was the chair for Native Plant Day, and she did an awesome job. That was her first time doing it. Although she helped a lot. She helped Jeannie a lot last year too. And we had over 450 people come to the event. And despite it being in a location we'd never been before, I think it was a success. I think Daniela's on a plane right now on her way to Europe. I also want to thank all of the board of directors, who I think everyone attended who was on the board Native Plant Day, the speakers, the vendors, the volunteers. I mean, that's what makes the Native Plant Day event so successful and so great. So I think we should have a big round of applause for all those people. For those of you who don't know, I do very little on Native Plant Day because I sell plants on Native Plant Day. So the only thing I did is I had to sign. I mean, there's a lot of bureaucracy. Go figure. with the county, it's gotten worse. All right. Some announcements now. So we have a field trip this Saturday, which I'll be leading to Emmy Thompson Preserve or Park, which is like on North Chrome between 8th Street and 27th, US 27. So this used to be like Melaleuca Park, and it's all gorgeous. The county did an awesome job there. And it's like a postage stamp surrounded by Melaleucas now. It is really amazing how that it can persist like that, despite being surrounded by Melaleucas. And they're becoming less and less. I did talk to someone at the park, Hillary Cooley at Native Plant Day, and she mentioned that they got rid of their last Melaleuca infestation in Everglades National Park just recently. So, yes, you have. If you take Chrome, like north of 8th Street, north of Dade Corners, and you're on your way to US 27, right before you get there, it's on the right. There's a motorbike track, and we're going to be enjoying the sounds of the motorbike track while we're there. So, but it's spring wildflowers. Even though it's a wetland there, it's not very wet right now. There could be things like calopogons, which is an orchid in bloom. Some of this, like the fleabane, should be in bloom. There's a lot of cool stuff that'll be in bloom there. Most people have never been to this park, so we at the Dade chapter like to go to places people don't always go to. So you should check it out. The instructions are in the newsletter for this month. Next month, we're having a field trip way up north to Jonathan Dickinson State Park and the Kitchen Creek Trail. Now, we haven't been there in 10 years at least. And this will be led by Dr. George Rogers, who is a retired botany professor from Palm Beach County. And he's a great blog writer. His blog is Treasure Coast Natives. It's one of my favorite plant blogs to read. You should check it out. It's on WordPress. He's just a wonderful writer, wonderful scientist who makes things very easy to understand. Again, April's a great time to see a lot of spring wildflowers. There's stuff at Jonathan Dickinson that's at the southern end of its range, things like Pogonia, which is another orchid that grows all the way up to New England. It's a terrestrial orchid. What else is up right now? The violets are up. The carfephoruses, the deer tongues are up. All these cool wildflowers are going to be blooming for us. And Kitchen Creek Trails, the trail that's near the river campsite. And so it's a 2-hour drive. So prepare for that. I recommend you carpool. That'll be April 25th. If I can make it, I'll go. Next month's program, we'll have Jeff Wazalewski from UF and IFAS. And I saw him at Native Plant Day. I think he might have given this program at Native Plant Day. His program is Hedges on the Edges, natives that get the job done. So if you're looking to plant a screen or learn how to plant a screen for your yard, you know, which natives make the best hedges, et cetera, you might want to attend that. I think Jeff is a Miami native. He's been here for a long time. He's been a member for a long time. I don't know if he still maintains his membership, but it's great that he's still around giving us talks. He's my age. He's not like ancient. Just a reminder, May is our annual members meeting. We will be holding elections, and we also have nominations for a nomination committee chair, which is Karina, who's our VP. She's, if you want to try and help out on the board, we do have a couple positions open. There's some important committee chairs, positions that need to be filled. Karina, you contact us at dadefnps@gmail.com. We do have opportunities to volunteer. We do have Patty Ferris and her group does the visitor center maintenance of the landscape there once a month or so. In the Everglades, the Everglades Visitor Center, the main entrance. And we do have, Scott has his program where we're doing conservation and restoration on some of our wild areas. We're still trying to figure it out a little bit. But there will be opportunities for that. If you feel like you need to get out and support your community, contact us at davefps@gmail.com. We'd love to have help. Ricky, is there anything new with the state? I mean, we have the executive director here, but nothing. Okay. Other than the dues are going up. Any other announcements? All right. We'll do the raffle descriptions. So we have a lovely assortment of natives here. And here we have a native Camacrypta. Which one is it? Oh, Nictitan. It's a hairy partridge pea. So Camacrypta, which used to be Cassia, is a partridge pea. They have these little yellow flowers, orchids flowers. They call that cystopinoid. And the leaves are just close up at night. They're not that sensitive to touch. They're just close up at night like our legumes do. It's in the pea family. So it'll have these little pods. It's a small herbaceous wildflower. And when I say herbaceous, that just means non-woody. Okay, it doesn't mean you can cook with it. And it gets about like that tall, has really hairy leaves. It is a host plant for probably one of the longtails. The yellows too? Okay. Sulfurs. All right, we got too many cups here. Or are these tick seeds? Those are, okay. We also have some of the real native tick seed, the stuff that's from down here. I saw that Patty Ferris brought this in. This is for our raffle. So if you want the local germplasm in your yard, which I encourage you do, ours has got much more narrow leaves than Coreopsis leavenworthii that's found further north. In Dade, we only have one native species of tick seed, which is Coreopsis leavenworthii. They took the whole genus and made it our state wildflower. Our state flower is practically endangered now, which is the orange blossom, as our orange industry is just completely folded. Real pretty annual wildflower. We have a wonderful lawnweed, which is native to cypress swamp, go figure. I don't know, I mean, you see this in cypressy areas or in wet swales like in the Keys, but it's also in people's lawns. This is a fog fruit. A lot of people call it frog fruit. That's not really what it's called. That's a corruption. The old timers used to call this creeping Charlie. That's what I grew up knowing it as. Another really good name is matchweed, because the inflorescence looks like those old school wood matches with flowers on them. So if you look at the flowers up close, they remind you of lantana. It's in the same family as lantana. And this is also a host plant for at least two butterflies. It's the white peacock and the Phaon crescent. What does the tropical checkered skipper use? No, never mind. What Mary's alluding to is she put together this awesome display on Native Plant Day, and go check it out before you go. You walk over there and just see all the cool lawn weeds. It's tropical checkered skipper. It doesn't matter. There's a tropical checkered skipper. No, pepperweed would be red admiral. It's okay. Anyway, check out those lawn weeds. I did a program when everyone was talking up butterfly gardens and they were fog fruit. So it is this. I thought so. So tropical checkered skipper is on the fog fruit. You can have more butterfly host plants in your weedy lawn than in a butterfly garden. All you have to do, and I gave a program on that because I noticed that everyone was tearing up their lawns full of natives. Because if you have a weedy lawn, It's probably mostly native. And you know, there's shocking things like, did you know that St. Augustine grass is a host plant to 6 different butterflies? That's inconvenient.
Yeah, but you have to let it grow.
You have to let it really, yeah.
For them to eat.
You have to look like the house that's like on the block that's no longer people living in it, you know. So, all right. So here we have back to the raffle. Here we have another host plant called Cheesy Toes, which is I never liked that name. Pencil Flower is another name. Although Cheesy Toes, someone talked me into it. It's like, you know, kids love that name because it sounds silly. This is another member of the legume family. And this is a host plant, I think, for the barred yellow, which is a little yellow butterfly that if you park at the light next to the, you know, the weedy field, you'll often see barred yellows there and dainty sulfurs. So it's a good lawn weed. We have another low growing plant, which is herbaceous. This is our native porterweed. If your porterweed grows like that tall, it's not native. The native one only gets to be maybe knee height. And this is found on the edges of pinelands. This is Stachytarfa gemescensi. There's another host plant, I think, for the buckeye, one of the buckeyes, and something else. And a really good nectar plant. It'll have this rat tail inflorescence with little blue flowers on it, and it blooms all year, so you'll always have flowers. This will also grow in partial shade. So for those of us that have filled up our yards with plants, you should be able to shoehorn this in. Anyone have a pond or a water feature? I don't see any hands going up. You guys got to put ponds in your yard, man, or a Tupperware pond or something. I mean, the poor insects, and it's a drought right now, man. What are they going to drink in your yard? Anyway, so when you get your pond, this is something you want to put in it. This is a native canna lily, a host plant for the Brazilian skipper. And this one has these big, gorgeous yellow flowers. Now you could keep this alive like in a saucer of water. It'll do fine. You'll need a bigger pot than this eventually. And you can see that it's rhizomatous, it spreads. So you could, when you get this, you could break it apart and create new plants. This is found in wetlands out in the Everglades. We call it bandana of the Everglades or yellow canna. Now here's a very cute plant, a little baby firebush. So if you feel the need to mother a plant and not stick it in the ground just yet, do not plant this in the ground right now. You'll forget about it and it'll die, or an iguana will find it and it's done. So firebush is a must for every yard. The native ones are usually a little bit hairy. I can feel this is hairy. If you have a firebush and there's nothing eating it, it's probably not native. If you ever buy, a lot of people have the non-native firebushes. Our native one is Familia patens. The non-native one is glabra, which means it's not hairy. Firebushes get big eventually, so that little guy will get like 20 feet tall, but you can cut it back every year. It comes right back. It's a hammock plant. It grows in the light gaps. Here's a pineland plant. This is a Carolina petunia, Aurelia caroliniensis. So it might be succulenta, but the flowers look too small. But I've been struggling with this too. There's a lot of stuff floating out there. Caroliniensis comes down to like maybe Broward, definitely Collier. Succulent is the more dominant one you find in Dade. And the flowers are usually like that big instead of like. This one hasn't had a good fall. No. It's in the Acanthaceae. It's a host plant for the malachite butterfly. Raul and I were talking about that earlier. Although if you want adult malachites, you need to have rotting fruit. So you have to have like a mango or something. All right, here we have a little baby wild sage, Lantana volucrata. This is a great plant for your yard if you have sun. It has little white flowers. You need little white flowers for all the cool little butterflies like atalas and other hair streaks, a lot of small skippers. But even the bigger butterflies will join the party on the wild sage. It's a nice shrub that gets about 4 feet tall. It's about maybe a little bit less wide, but around 3 feet wide. It's a short-lived shrub that is, probably live about 4 or 5 years. And like, I'm done. So it will spread in your yard, but not aggressively. So you might get one pop up here and there. Yes. No, I'll get that later. All right, here we have a nice wildflower that I've noticed it's becoming less common in the wild. This is forked blue curls. And I don't know if this is a climate change or something, but I imagine it probably doesn't like it as hot. You find this in coastal strand habitat, like on Key Biscayne. There's a few pinelands. Yeah. So on Key Biscayne, it used to be more common in some pinelands. You don't see it as much anymore. This is a member of the mint family. It has these little blue flowers. Probably a bee pollinator specialist. Most members of the mint family attract bees and their hymenopteran cousins, like wasps and stuff. Another member of the mint family, someone brought in our native endangered wild basil, which is Ocimum campechianum, very fragrant leaves. This is a short-lived herb, but it will spread in your yard if you have the right conditions. Bare soil, it's a pine rockland species, although it also grows in openings of hammocks. Very rare. I've only seen this native to 2 or 3 wild areas, and it's historic to Big Pine Key. I'm searching for this on Big Pine Key and Cactus Hammock to no avail. It hasn't been seen there since the 90s. Here we have a trio of little pinklets. So this is a member of the Acanthaceae, a cousin of that Ruellia. Has little pink flowers you can see here. This grows in the transitional zone between pinelands and prairies, but it grows fine in your pineland. You want it in a sunny area, but it'll tolerate low levels of light sometimes too. The sunnier it is, the more it'll flower. So that's a great little wildflower that you can't buy. You only find it here, folks. Here we have a shrubby Eupatorium, or shrubby thoroughwort. What a horrible name. Coinophyllon villosum. And this is a great, remember the Asteraceae, it's a shrub. It's found in southern Dade in their pine rocklands there. It's not in the Keys. It's just in that weird area of southern Dade, like Tetrazygia is the same way. This will produce white flowers, and it gets full of pollinators, just like some of those other shrubs we talked about earlier. Okay, here's our tick seed. We already talked about the seeds. Here's the plant, state wildflower. This is an oddball that Raoul's grown. Fairchild started growing this. This is a purple top trident. It's a native grass. It's rare. Grows on the edges of hammocks. Real attractive inflorescences that are purplish. This is actually not a bad landscape grass to introduce. It just hasn't caught on yet. The inflorescences get about that tall. The leaves are about maybe 2 feet. And for those of you online, that tall means about 4 feet tall. All right, here's Elliott's aster. This also grows at a big cypress in some areas of the Everglades. I haven't seen it very much in Everglades National Park. It's all over off of like Loop Road. And this is one of the bigger asters that gets about 3 or 4 feet tall. The inflorescence will. Big, composite flowers. Asters are fun. They're beautiful. This is great for like a meadow garden or a butterfly garden or a pine rockland or something like that. It's not really a pine rockland plant, but it has the same conditions of full sun that you would want. Here we're getting close. Blucia odorata, which is a common name is camphorweed. And this sounds horrible. The leaves are very sweet scent is another one. So the leaves are aromatic when you crush it, hence the name odorata. It's another member of the Asteraceae. This grows in disturbed soils, like in coastal areas, coastal freshwater wetlands, or interior freshwater wetlands. Like in the hole in the donut where I work, this comes in and goes. It doesn't stay. It's ephemeral. But real pretty pink flowers that pollinators will visit. It's an herbaceous wildflower. It doesn't get very big. Here we have a seaside gentian. This is one of the things that usually blooms this time of year with real pretty purple flowers. It's a native gentian. This is another one that you'll find in like coastal wetlands. It's also in interior wetlands. Real pretty wildflower. It'll grow on roadsides. Here we have our rare endangered Ipomoea tenuissima, which is the hairy little pine rockland one with pink flowers, has these heart-shaped leaves. So this is, I think in Florida, it's only found in our pine rocklands. I don't remember if this is in the Keys or not. I'm pretty sure it's only in Dade in the Bahamas. And toward the end, We got our native violet. Now the violet peak flowering, it starts in February and continues in March. There are still flowers. The flowers become cleistogamous in the summer, which means it blooms, but it doesn't have petals. It's not showy. But what's nice about this viola is it grows in shade. So a lot of us already have full yards and you're wondering what will grow in shade and you're trying to replace your lawn. This is a really good one to mix with other plants. I don't think you'd ever make a full lawn of it, but it's a nice wildflower. And it's a host for the variegated fritillary. Yes. I've had it come up in my lawn and I don't water my lawn. So no. What's that? Yeah, it'll be happier. Where I don't know where the provenance of where this originated. She got it here, but that doesn't mean someone got it from somewhere else. Don Keller found a population outside of Massahammock, and I revisited that population. It's in someone's yard, and that population all had white flowers. I've never found blue flowered violas in Dade County. I go to Big Cypress, they're there, or you go to Palm Beach County and they're there. We got other things here that aren't set up yet. I don't know if those are for the raffle or not. Okay. But that's it for the raffle. I know it's 8, but I want to say 1 or 2 words real quick. I got this really cool handout on native plant pollinators. Karina, do you remember or Ricky, do you remember where this came from? Someone left it at my, is that your alls? I just noticed this winter that I don't have many bees in my yard right now. And we've had all these mangoes and I just want to remind people that the drought is hard, not just on your plants, but on the critters. So put out saucers of water. Bees, even insects drink water, and avoid spraying your yards. You don't need to mow your lawn right now. You can tell the guy who's mowing your lawn to take a break and stop mowing your lawn right now so that the wildflowers, even if Biden's comes up, those are great help for the pollinators. The other thing that we have a tendency to do is we don't like bare soil. It looks poor, it looks bad. A lot of bees nest in bare soil. So be sure to keep open patches of bare soil. And the last thing I'll leave off with about my pollinator thing is don't trim all the dead stuff off your plants. A lot of bees nest in there. Try to avoid killing paper wasps. Most of them aren't going to hurt you. The only reason why you get stung by one is if you took your hand and did this. I mean, I've had one above my patio door for years and years, and I'd walk in and out of that door, never been stung. And I'm a big goofy guy. So don't be terrified of all these things. There will come a day where they might be gone. And you'll be like, man, I remember these used to be everywhere. We used to talk about land crabs. Oh man, those used to be everywhere. Or lumber grasshoppers. Oh man, those used to be everywhere. And now there's man, those are gone. So anyway. All right, we have a great speaker, a wonderful speaker, Melissa Fernandez, who's going to give a program, Protect Our Wetlands, Local Action, Statewide Impact. But I'll give you a little bit of bio for Melissa. She's a scientist, a lifelong educator, an environmentalist who serves as the executive director of the Florida Native Plant Society. She's Florida born and raised in Miami-Dade. Her work is rooted in a deep personal connection to the landscapes and communities of the state. Trained in environmental chemistry and pathology, Melissa brings a big picture perspective to conservation, connecting ecological health with community well-being. She leads statewide efforts to protect Florida's native plants and biodiversity by strengthening community-led conservation, partnerships, and on-the-ground work across Florida. Please, let's give her a warm welcome.
This is too fancy.
Hi, everybody. Hi, it's nice to meet you all. My name is Melissa Fernandez de Cespedes. I like to start by letting people know what my name means because it's very topical, especially after Steve's explanation about pollinators. So Melissa means bee, which is really fun. And the Cespedes means of many lands, lawns. Cesped is Spanish for grass. So that's pretty cool. I think it's pretty fitting for this role I have. I don't know if I'm still the new executive director. How long does a person need to be in a role to be new still?
Probably the first year.
All right, so I'm past that. So I'm going to be talking today about some of my master's research findings. My master's work was here in Dade County. I was born in South Miami Hospital, I believe. I don't know. And I grew up in Hialeah, Miami Lakes area. My master's and the work I'm going to be talking about is work I did while I was an FIU student. So I'm not a gator. I am not a hurricane. I am a panther. And proud. And I was working in the environmental sciences department. My mentor was in the chemistry department. And it was one of the best years of my life doing this work. And I went off to go do other stuff. And I kept feeling a calling to come back to Florida and find my way into a professional role in conservation. So I feel very grateful to have this position and to work alongside all of you in protecting what's precious. So shown here is a manatee, one of our favorite animals, and it's floating over a seagrass bed of Thalassia, which we also call manatee grass. And I'm showing this because my master's work was in seagrasses. Oh yeah, there you can see it. How do I advance? Okay. And so before I start, I wanted to explain to you all why I love FMPS so much, why this position, the minute I saw the job posting, I applied like that day. Because it's not just about plants, it's about the people. As a society, we're a community, and that's what makes this really fun. One of the things that I've found is I feel very alone and helpless when I see what all this development has done to our wildlife over the course of my lifetime. And I didn't know that there was a group of people that felt the way I did. And you can ask my parents, I was a little planeteer and I really wanted to save the planet, save Florida. So it's kind of like a lifelong dream to be here and to be with all of you. Let's see, which direction am I going? Is it gonna play on its own? And so I'm gonna share a little video with you. We posted it on social media. It really, I don't know if the audio is going to be loud enough, but I love this video. Charisse, our operations director, put it together, and it's celebrating FMPS. I don't know. We'll figure it out.
I have to click out and then get back.
Yeah, you can. Can you play it?
Yeah.
Sorry. Well, at least it's gonna play. That's always the struggle with videos and like slides. I like to create a challenge for whoever's hosting.
1,000 people every day are moving to Florida. The state is changing so rapidly. Plants can't run away when the bulldozer comes. All the stuff's connected together somehow. Individually, they're
change. We have people earnestly striving to get that done.
You might see some of your friends in these images. And the audio was Craig Hugel, Roger Hammer, and Peggy Lance. Some really big hitters. It's not moving. See, I love a challenge. So I just love that video. It's on our website on the main page because I can't get enough of it. So, so like I mentioned, I'm a Hialeah girl. And growing up, the Everglades was my backyard, which is really special. I remember being in Shark Valley. I remember going on Hinga Trail. I remember going with my friend and her mom to go photograph the stars out in the Everglades because it's nice and dark out there, or was. And so the best year of my life was this year doing Everglades field work and working in Biscayne Bay. And that's me with my lab mates having the time of our life, truly. And then moving on, I've gone away. I worked at the NIH and then I came back. And now I get to botanize with coffee with all of you. And that's the best job ever. Oh my God, I'm going in the wrong direction. All right. So Florida is a biodiversity hotspot. We have a lot of native plants and we also have a lot of naturalized plants. And we have a wide diversity of ecosystems. And South Florida is really heavy on the wetlands. So we're talking about wetlands today. Yeah, I keep going in the wrong direction. All right, so I love this image. I also hate it at the same time. It shows the coverage in blue is the wetlands in 1784. And then on the right-hand side is 40-ish years ago. This makes me sad, but I think this is a really good representation of what is happening to our state. And this is from Priceless Florida. It's a great book. I highly recommend. Yeah. And I like to underscore, like, why plants? Everyone's like, why plants, Melissa? That's like, why? I don't get it. It's because they're the foundation of everything. So they're right at the primary producer step. So we talked about how pollinators need plants and host plants. So without plants, really there's no life. And that includes for us as well. So I'm going to go into some science. I do not expect everyone to understand statistics. I'm going to kind of like glaze over the stats and just give you the takeaway. But it's also a great example of the kind of work people can do for their graduate studies and how I want people to understand that they too can study plants. So my work was on what we call submerged aquatic vegetation, and that encompasses marine angiosperms and macroalgae. And I was looking specifically in the lab. So I would get samples out in the field. I was looking in the lab for herbicide content in the water, in sediment from the bottom underwater, which is my biggest fear, is going underwater. And seagrasses and macroalgae that I collected that I was able to find. And most of my work was in Biscayne Bay. So seagrasses, we always think of manatees and turtles, but they also are important. Sea life in general, plants underwater are important. here's an example. And this is for Thalassia testudium. So very specifically, the Thalassia that grows in our waters. And they have found that there is, there's molecules in these plants that can be used for human health, such as anti-cancer. And one of the things I learned at the NIH was they actually have these huge panels of chemicals that they've extracted from plants, especially sea life. And they test this against viruses and against cancer cells to find, in big screens, to find new novel compounds to treat disease. So again, another reason why plants are important for us. So this is the paper I published. My mentor, Piero Cardinali, he was wonderful. And I'm going to go into some of the highlights from this. I keep going the wrong way. So what I did was I studied herbicides, specifically, and I do not like laser pointers, specifically ergorol and atrazine. So ergorol is an anti-fouling compound. They used to put it, it's now banned. They put it in paint for submerged structures to prevent the biofilm from forming and then other life attaching itself to those structures. And then atrazine is actually used commercially, and that's why I have a little like farming thing here. And people also use it in their landscapes, and it all leaches into the water. So it comes off, it leaches out of the boat paint or submerged structure. Atrazine leaches into the water after being sprayed. And the question is, how much of it is out there in the waters that we're swimming in? And also, do seagrasses and other submerged aquatic vegetation, do they bioaccumulate those compounds? Because the way that these herbicides target the seagrasses and broadleaf plants is it binds to proteins that are involved in photosynthesis. So it's just blocking photosynthesis. So these are the chemical structures. They all are very similar. That's the takeaway. And I have some information about the different compounds. So ergorol is the anti-fouling compound. I am very happy to report it was banned in the US. It had been banned in other places well before it got banned here. And so as of 2023, it's been phased out. One of its metabolites, which I refer to as metabolite 1 or M1, is a degradation product of ergorol. And ergorol is generally degraded by UV. And there's also microbial life that also can degrade these compounds, but not very effectively. Atrazine, it's not banned, unfortunately. So it's still being used. I'm gonna guess it's not banned because of, farming lobbies. Yeah, oh yes, thank you for, I have it right there. It's an endocrine disruptor. So it actually affects the reproductive development of amphibians and other small life. And it's everywhere. I detected this everywhere. So I just wanted to talk a little bit about algae and seagrasses very briefly. What is the difference between them? They're both under the umbrella of submerged aquatic vegetation, but they're different. So algae will adhere to structures, and seagrasses have rhizomes and roots, and they actually stabilize the ocean or river floor. So without seagrasses, you have very turbid water, and the water quality declines. And it's a self-feeding loop wherein once the water starts to get cloudy, it blocks the sun. So the sun can't reach the seagrasses, so they can't photosynthesize, so they die, and then it gets cloudier and cloudier. So dredging is terrible. That's the takeaway from that. But what's really interesting, because we always think about land plants flowering, they flower. They flower. And it must be pretty special to see that. I did not. Every time I was going underwater, I was panicking and I didn't see any flowers. I have a very strong fear of drowning. So some of the submerged aquatic vegetation that I found was the Thalassia testudium, which is the turtle grass, Halodule rhydi, which is our shoal grass, and the Syringodium filiforme, which is our manatee grass. I also saw a series of different macroalgae, and so here's 2 examples. I didn't see them a lot where I was sampling, so I would get like a few samples max. It wasn't like a great amount, because where I was sampling had a lot of human activity and there wasn't much life actually to sample. So you can ignore most of this. This is all to say that in this study, I was comparing what I was finding locally to other scientific studies that had created benchmarks. So how much ergoterol is toxic to corals, to plants themselves, actually, how much ergoterol kills plants. And I use these values throughout, but you don't have to memorize them because what's important is the findings. So this is where I did the sampling. This might look familiar. This is why I wanted to talk about this, because these sampling sites are here. So I was studying South Florida, Biscayne Bay region. So we have, like, this is a zoomed out photo. Key Largo Harbor, Chicken Key, and Coral Gables Marina. Zoomed in, you see Chicken Key, and that's the spot where I was sampling. It's beautiful. I don't know if people like that, but the seagrass beds were beautiful when I wasn't panicking. Coral Gables, I sampled a bunch of sites there. That was disgusting. The water was gross. There was turbid. The sea life was nearly gone, nearly absent. I'm pretty sure by now there's nothing there. And then here's Key Largo Harbor. So this is a series of canals, and we sampled outside of Key Largo Harbor, went into the canals. And so this is where there's the least amount of water turnover. And I actually ended up finding that that was where we had the most ergoterol present. Was the more the less water turnover, the more that molecule was there. And I also, in those canals, because it's turbid and there's boats and stuff, there wasn't any seagrasses. So I was collecting mostly out here. Yeah. Which to me is open water, and that's terrifying. And then also the Miami River. I was sampling in the Miami River back when it was not quite dead yet. And then after this study, I think they announced that it died, like the dissolved oxygen was just not sustaining life anymore. Huh? No, but I did put my arm in there to get the samples. Ugh. I also saw a lot of stuff. I saw stuff that I don't think is legal, where people were sanding boats and all the dust of the paint was falling into the water. And I'm like, but that all has ergoterol in it. So like they already made it like microplastics kind of, so they made it small so it's easier to leach. It was pretty bad. So this is what it looks like to collect samples. This is a lifetime ago. I'm happy on the boat. I'm panicking in the water because I have to dive down. It's about 10 feet, or maybe 12 feet. And I don't have a really good set of lungs to like really hold air. And there's a lot of panic that the boat's gonna move. And it did happen. So from the surface water, so here, this is the surface water. I just stick my arm in with a container. We let it fill up with water. We cap it. That's easy. I put it in a jar. That's hard. And then I have to find the seagrasses, which in Coral Gables and in Key Largo Harbor were not plentiful. So I had to kind of snorkel around. I had the worst sunburn on my back one time because I did not have the appropriate swimming shirt, which I ended up getting eventually after that sunburn. Don't tell my mom, she's in the back. Ugh, you learn the hard way, right? And so you can ignore logs. I know that makes people panic. But I took all the concentrations, I measured the concentrations of these herbicides. So this is showing just for Igorol, because it's the one that was really the focus of my study. And I ranked them and plotted all the samples. So there's all the water samples. In this is not only the years that I sampled, but years previous that my lab had sampled at these locations. And the important thing here is this 90th centile or 90th percentile, depends if you're in Europe. They say centile. And the 90th centile of Igorol was well above the amount that's been determined to kill or affect corals and other small, like phytoplankton, small sea life. So that's bad, right? So the majority, the amount of Igorol that was in that water was directly impacting marine life. And we have a coral reef, so don't forget. It is a little further out, so these concentrations were going to be diluted by the time that they reach the reefs, but it doesn't help. Yeah, I'm probably used to. So this is splitting out the data by regions for M1, the metabolite, and this is just showing that there's way less of the metabolite in the water. The amount of M1 that you would need to affect sea life like negatively is really high, so that was not the risk. It was really just Igorol. I keep going the wrong way. All right, so now I'm collecting seagrasses. So this with the shirt this time. I bought this shirt after that sunburn. I'm definitely panicking here because my head isn't even in the water fully. I'm like, and then on the right, I have my collected seagrasses wrapped in aluminum foil that's been treated with hexane, so it's all sterile and non-contaminating. Put it in a Ziploc, put it in a cooler, take it back to the lab. So now we're looking at the data for the different plant life that I was able to find. Obviously, I found a lot of Thalassia. That was a lot. But in terms of their bioaccumulation of Igorol, so I'm measuring how much Igorol is in the plant tissue. I did find it, as you can see. And bioconcentration factors greater than 1,000 indicate that the plant or life, whatever life you're measuring, is efficiently selectively bioaccumulating. Because remember, it binds proteins involved in photosynthesis. So what this is telling us is that seagrasses and other submerged aquatic vegetation can act as sentinel indicators of herbicide contamination. And there's differences between the species as well, and that's why I split them out. Okay. Yes, I went in the right direction. Okay. So I did the same ranking here, and the 90th centile for Igorol was great. It was over 9,000, nearly 10,000. So again, that's like really bad. It's well above the amount that is, going to kill sea life. And M1, it didn't really efficiently bioaccumulate. So the risk is really the main compound, the parent compound. And then I had fun. So I thought while I was out there, I was like, okay, we're measuring tissue. It's, what we can get. What if we look at different segments of the plant, and will there be differences in the organ that I'm measuring it in? And then furthermore, because I like to complicate things, what if I compare two species? And what I found was that for Halodule, the bioconcentration happened most efficiently in this region, in the meristem, whereas I really didn't measure any in the roots, which is interesting because I was able to measure it in Thalassia. And there are differences along the Thalassia leaf in terms of its ability to bioaccumulate. I don't really know why. I'd have to study how the leaves are growing and at what rates. But this is the beginning of a new scientific question. All right, so next I did a transplant. What if I take seagrass that has bioaccumulated igorrhea and I transplant it to a site that's pristine, does not have igorrhea? Will that plant depurate or like release the igorrhea? Would the igorrhea levels decrease in the plant tissue? And vice versa, if I take seagrass from a location that's pristine and I put it and I transplant it into what ended up being Coconut Grove Marina, what are the kinetics of it bioaccumulating igorrhea? I keep going the wrong way. So that's the transplant device. It's fancy. It's just a PVC pipe and we cut some holes in it. And then in the hole in the top, we put a little like color tape so we can identify it easily. And so this is just a schematic of how this worked. We have our transplant, our control, and our indigenous control. So we were controlling for the transplant device itself, and we were comparing that to plants that were not disturbed locally and the transplant as well. It's very simple. The plants literally we just take a plug and pop it in. And what we found was that, and this is what I was working with what I had. So Hollow Dule from Coconut Grove transplanted into Chicken Key. It depurated the igorrhea very quickly, within a month. For Thalassia, I transplanted Thalassia from Chicken Key into Coconut Grove Marina, and the data was a little funny, so we plotted it 2 different ways. Either which way, it does bioaccumulate igorrhea pretty heavily within a month. Okay, up or down? So these are the transplant devices in situ. You can see the flags, and we use different colors to indicate, what's what. And then we had a cooler where we kind of stored them while we moved them from one location to another. It was a very big day because you want to do it that day. So there's a lot of conclusions that are scientific and heady, but in essence, the seagrasses are bioaccumulating igorrhea. They're bioaccumulating, depend on the species, in different parts of their leaves and or rhizome systems, and they can serve as indicators of herbicide contamination. So what's happening is in the water, it's very diluted. But when you concentrate it in the leaf structure, you can measure it much more easily. And then I wanted to do an invasive seagrass alert. So about the time that I started this job, I went to visit my mentor, Piero Gardinelli, Dr. G, and we were having a chat, and I was like, so how's the seagrass stuff going? And he said, there's a new invasive. They've detected a new invasive seagrass. And we think a lot about invasives on land, Brazilian pepper, Melaleuca, et cetera, but we don't really think about invasives underwater too much. We think about fish, but not so much about plants. And so I found an image of the invasive Halophilia, and it looks very different, thankfully. So I think it's going to be easier to monitor, but they're finding it right where the Everglades water just kind of comes out into the ocean, that area. And they think it came from a ship transporting something, and it had probably attached itself at port somehow, and I don't know, transplanted it. It seems incredible. And so that was my research, the highlights. And I also have some findings from the Everglades Coalition Conference. I wanted to summarize it. I don't know how much time I have. Oh, okay. So this is the photo I took in that same time frame of my life. That was a lifetime ago. This was at Shark Valley. And here's my favorite little alligator. I should have worn my alligator earrings. So in January, I attended the Everglades Coalition Conference. We are looking for a liaison who would like to take that role. We haven't heard back. It was fun. I will say that. I saw familiar faces. The musician that was at Native Plant Day was there. Kim Heiss was there. Lots of fun people. Hillary Swain, obviously the ED for Friends of the Everglades. And there were different sessions I attended. I summarized them. I'm gonna put a blog post together that's also going to summarize what I heard there and learned there. And so for the different sessions, I kind of put some key takeaways and some calls to action. There was a lot of discussion on SERP, on the ESA, how it's been violated. For example, there was a whole discussion on Alligator Alley. There was representatives from the Miccosukee tribe that were speaking there. I was in good company, essentially. Everybody understood the assignment. So they talked about how that land where they built Alligator Alcatraz is spiritual land. They are really upset, not only for what's obvious, but because not only because of the environmental impacts, but that's their land. They have rituals, they use the plants there, and now they don't have access to their land. They were never consulted. That is legally, they're supposed to be consulted first, and obviously we know that there was not any environmental assessments before they started construction. And so this sets a very dangerous precedent on our conservation lands. And so thankfully, the Friends of the Everglades is challenging this in court. They're full on going after this. And so the call to action for us is to stand with the Miccosukee tribe and uphold these regulations, these federal regulations. And moving forward, just defend tribe sovereignty and their right to their land. They know how to manage their land. They know what's out there. There's a lot that they know that we don't know. And we can't lose that knowledge. There were also discussions about Lake Okeechobee. So I don't know who heard recently that Lake Okeechobee was like the most polluted lake.
It's up there. It's definitely up there.
Google it. I was floored to hear that. I'm like, why? We're doing all these restoration projects. Why is Lake Okeechobee still and getting worse, actually? So what we need to do is support conservation land corridors, push for smart growth. What is smart growth? I'm in Gainesville right now. I'm so sorry I left you all. It's cold up there. And the plants are different. So there's a lot of condemned houses and buildings. There's a lot of property that's kind of being squatted on by the landowners, waiting for a developer to come in, pay an exorbitant amount of money to build an apartment building. If those properties are kind of sat on, then we have urban sprawl. So smart development is building up where we've already developed. So that's what we need to push for with our communities, our legislators. And it's called infill development. I just learned that term, but I've always been wondering, like, why is this just sitting there? This is so upsetting. And instead we have to buy a car, drive down the road forever to get to the location we want to go when it all could have been built locally. So this is more about smart growth and urban sprawl. That was actually a big theme for the conference. And so the call to action here is incentivize building in the communities we already have. So when we talk to our legislators, that's what we're pushing. Another session was on watersheds in general. Are these management plans working? So this is where it gets tricky. And this actually relates back to the atrazine of it all. We're still detecting, so these farms are saying they're complying with regulations. But when we're out there in the field sampling in Big Cypress and in the Everglades, and obviously in Chicken Key, which is Biscayne Bay Preserve, a preserve, we're supposed to preserve it. Why is there herbicide there? We're detecting way too much pesticide and herbicide. And that's a problem. So they're saying that they're abiding by these plans and regulations, but what scientists find don't line up with that. And so we have all these herbicides, native plants can't recover because it attacks their photosynthesis, and our water quality does worsen because we have all these chemicals. And those chemicals do eventually percolate to our groundwater, and it's everywhere. It's everywhere. So we need to demand science-based water policy. We need to tell our elected officials we want clean water. We don't want to drink herbicides. We don't want endocrine disruptors in our water. And we want this to be enforced, not like self-reported system. So more growth management, tying that with conservation policy and different legal tools. So on paper, we have comprehensive plans as one tool, the urban growth boundary, which I believe keeps moving, and litigation, which is what Friends of the Everglades is doing. And the reality is these tools work slowly and only if we use them. Counties don't tend to enforce the boundaries, the growth boundaries. They are either not knowledgeable in the things that we are, which is, hey, we're destroying wildlife and really sensitive native plant communities, because they're seeing, oh, this is going to create jobs and this is going to improve and develop our communities. And another problem, and we're seeing this with Friends of the Everglades, is litigation is slow. It doesn't happen fast. And by the time it gets litigated, the damage is done. And a lot of that can't be reversed. So we need to demand enforcement of our comprehensive plans. We need to challenge our community leaders on maintaining those growth boundaries and stop sprawl. And then this is where our local artists get a shout out. They had a panel, Kim Heiss is on it, like I mentioned, and they talked about the art residencies that they did in the Everglades and how special that was, and how art itself connects people to these important conversations, because sometimes it can be heady, like the science, right? What does that mean? How am I supposed to feel? And that's what art gives us, is a feeling and a meaning. So the call to action here is partner, and you're already doing it. Partner with local artists of all kinds. Really impressed with what Dade has done. The t-shirt that I bought at last year's Native Plant Day is so special, and Kim signed it. So I'm like, really, like, look who signed it. My mom got one. She got it signed by Kim. Like, I'm more connected because I have a little piece of that art with me. At the statewide level, we've been discussing murals, creating murals that educate about our endangered plants, especially Dicerandra, Torreya, Warrea plexifolia. And we're going to start working on one of the rhododendron chapmanii. We're working with the Atlanta Botanical Gardens to start a project in that field. So for chapters in general, that they can just highlight their artists in their newsletters and at their events. For y'all specifically support her, support Kim, and other artists who translate botanical science into very compelling artwork. And I would just want to let you all know, you're the conservationists. You're the ones that are making this happen. Your voice influences the decisions that are being made. So we do have power. You know, I was a child and I was watching the development take over Miami-Dade. The Palmetto was under construction forever. Now we have all these express lanes, and I just see more development going westward into the Everglades. So you're here. Get to know who your leaders are, your commissioners, your representatives, your senators. We had a very, very dramatic legislative session just now. It just ended. They did not pass a budget, I believe, and so they're going to have to reconvene. But there are some pretty gross bills that were on the table. And we didn't win all those fights, but we are a heavy influence on what's happening in Tallahassee. So back to development. This is our current land usage in orange throughout the state. It's projected to be pretty bad in 2060. Good news is there's no longer 1,000 people moving to Florida every day. That number has decreased. Yay. And I think it's like 100. Yeah. Yeah. Really importantly, with this development, the Florida scrub jay, our favorite bird, should be our state bird. Their habitat is going to shrink to the square footage of 3 times the size of Manhattan, which Manhattan is really small. It's really small. So here are some photos taken by Ethan Liu during the Protect Our State Parks rally in Tallahassee. These are just people like you. There was kids there, adults, college students. It's beautiful to see the wide breadth of diversity in humans trying to protect our biodiversity in Florida. And so this is my call to action to you. If you're not signed up to receive action alerts, please sign up. You don't need to be an expert. It's on our website. Every single page on the website has a sign up right in the footer. You can't miss it. We also have our legislative priorities up on the website. We post the letters that we send to our senators and other representatives. We're advocating for Volusia Forever. We're advocating for Florida Forever. We want at least $300 million for Florida Forever. That's the bare minimum. And they are probably going to give it $35 million or less. And Florida Forever is really important because Florida Forever conserves land. Once it's paved, we can't do anything with that. So we want to make sure it's not paved. And we do also support rural and family lands, but Florida Forever is getting cheated out of what it's supposed to be getting. I also graphed the Florida Forever funding over time. Take a look at that. You'll get real upset. And then lastly, I want to end up with some highlights from FMPS. Last year was our 45th anniversary, which is the sapphire year. And I created this collage of the closest color to sapphire I could find in our native plants. So 45 years, that means this year we're 46 years old. That's a long time. That's a long time of conservation. We should, and Dade is one of the first chapters, I believe. Yeah, congrats. Yeah, keep it going. So there are other ways you can have an impact other than harassing your representatives. You can join at the local level. I see Ricky's right here is a great example. She works with y'all, and then you can also contribute at the statewide level. And simple things like action alerts. Sign up, act fast. When we give those alerts, And we are very selective in what we're alerting for to not like not inundate you with them, but sometimes they're bad. They are voting on stuff in real time. So it's like an email that you're like, oh, I'm busy, I'll do it later, and then you forget about it. In the moment you see the email, you need to know who your senators, you need to know who your representatives are. You need to send them an email, kindly explain that you are a constituent and that you oppose this bill because XYZ. It's initially intimidating. I've never really talked to my representatives, but now I know who mine is, and I know all the bills that they support. Another thing you could do is volunteer for a land management review or mentor a new reviewer. These are important. We are helping our local land managers inform how their plans are, if they're working, if they're not. And I've heard that we can give some advice off the record. Right, Ricky?
You can endorse bills. You're not allowed to endorse politicians, but you can talk to them. It's not like you can talk to them. As far as expenses, the law, the rules for lobbying is only 5% of your expenses could be used.
Oh yeah, no politics, only policy. But for land management reviews, we get to go out and we essentially kind of grade. I haven't been on one yet, but I will be attending one soon in Polk County, I believe.
I've been on one. I think Scott, you've been on one. Land management review.
And one of the things we're going to start encouraging our chapters to do is get to, if they don't already, get to know your local land managers, because they're struggling. Funding's cut, resources, employees have been fired, so they need help. And this is one really tangible way to have a direct impact on native plants, is to find your land manager, find out what the need is, and help fill that gap. And then the last, I'm going to end here on a really positive high note, a celebration. Our 2027 conference is in Daytona. It's going to be hosted by the Pawpaw chapter. If you want to volunteer or donate to the silent auction, please email Athena Phillips. She's the conservation chair and the VP of administration at the statewide level, and she's really nice. She doesn't bite. And if you want to get involved, we have a lot of places for you. One of the things I like to say is it's a choose your own adventure, right? You can have it your way. There's so much to do in relation to native plants. Our pillars are science, conservation, education, policy and advocacy, and community. So those are the 3 big categories. And there's a place for everybody in that.
About the conference, who's your keynote speaker?
We don't know yet.
Okay.
2027.
Oh, this is 2027.
Yeah, it's,
so there's not one this year.
no, it's biennial now. Yeah.
Is it still going to be bookended by field trips
Yes.
on Thursday and Sunday?
Yeah.
And then the conference activities Friday,
Yeah.
Saturday?
More or less the same schedule that it, yeah.
And when's the last time you've had an in-person conference?
Last year.
So you're having it every other year now?
Yeah, biennial. Yeah.
Look at.
I didn't do that.
I've not been to an FNPS conference.
It was the most fun.
So they're a lot of fun. Field trips go to some really spectacular places. This one's on the beach. So
Yeah.
it'll probably be in a nice hotel.
Yes.
And
Yeah. It's
there's some cool nature up that way. That's up near Jacksonville.
I think one of the field trips might be the Enchanted Forest.
Yeah.
Yeah. They the hotel where it's being hosted is right on the Atlantic. There are bonfires there. It's really, really beautiful, actually, the location. And the conference rooms, the big rooms, there's some that have floor to ceiling windows just facing the ocean. It's beautiful. And if you're not facing the ocean, you're facing the intercoastal. So you're,
It'll be in May still, right?
yeah, April. Yeah, first week of April. Yep.
Well, thank you, Melissa. We still have time. Questions?
I have more slides.
Go ahead, Gwen.
As soon as you said murals, and you keep mentioning these Central and North Florida listed species,
Mm-hmm.
I started thinking windwood and ghost orchids.
Mhm.
We should have a mural down here.
Yeah.
We got plenty of federally endangered dingleweeds.
I'll tell you where I think I drove from Orlando today, and I stopped off at Fort Drum Plaza. Sorry, Dad, I took the turnpike. You got tolls. And I saw some mural-like artwork on the walls. I took a photo of it, but it was pro-environment and conserving our lands, pro-Everglades. And I said, "Oh, a rest stop. That has high visibility." Yeah. Lots of kids.
So does our basil.
No. I remember you from the conference. You were there last year.
Yeah.
We did the bat houses.
Yeah.
Yeah.
Other questions?
I hope. I hope. I don't know. That research was done in 2007, 2008. I defended it in 2010. I published it in 2015. So I have no idea. Yeah.
Could you tell us about your staff?
Oh, yeah.
Who does what?
So I'm the executive director. I do 20 jobs. You guys got me for a really good deal. And my right hand is Cherie Smithers. She's the operations director. She's the person who answers the info at FNPS emails. She creates all the membership reports that you all use. And she helps with all of the operational stuff, keeping our organization up to date on all its filings and all the deadlines, et cetera. She is a very organized, data-centric person, which is perfect. Yes.
She's also the one that's heading the shift of the whole database.
Yes, she shifted our member database and has been sanitizing the data, which means removing duplicates, updating contact information. If you have a membership issue or concern or trouble with any of your data in our system, she's the one to talk to. And
all be done by volunteer.
yeah, it also, yeah, we were purely volunteer led for a long time. Yeah. So having staff is a newer transition. You all know Valerie Anderson. She's the communications director. She is actively having a baby right now. I think its name is Jasper, and it's entering the world sometime in the next 72 hours. She does the social media, the action alerts. She does a lot, the newsletter, the Stable Miner. And then we have Lily Anderson Messick. She's up in the Panhandle, and she primarily does the organization of the land management reviews, the Torreya taxifolia work. We're trying to save this endangered conifer that is essentially functionally extinct because it cannot reach sexual maturity due to this fungus that keeps killing it back. So we're working with the Atlanta Botanical Garden for that. She's incredible. She's also our like resident botanist. So if I'm like, hey, what's this plant? Can you identify it? She'll help me with that. She's really good. And I even sent her a picture and I was like, is this a queen butterfly? She's like, yeah. And I was like, oh, thank God, yes. Taxa Tuesday, yes.
On the YouTube channel.
Yeah. And on social, yeah. And that's the four of us. And
So are you guys spread throughout the state pretty much?
yeah, Lily's in Tally. Valerie is in Polk County. And me and Sharice are in Gainesville by accident.
FNPS now has conservation.
We've had it for years. We leased it to Putnam Land Conservancy and they've been doing the restoration. So we have 14.4 acres around Oklawaha area. And the purchasing of that was a complicated mess that I'm trying to figure out, but we own it. And Putnam has been clearing the land to open up the tree coverage, because what we're trying to do is create the appropriate habitat for the endangered Florida sand skink and for Warrea amplexifolium, which is a beautiful annual plant in the mustard family that requires sun. On that property, I did see an active gopher tortoise burrow, and they're gonna do a fire this winter. They're at the level where they can now do a prescribed burn to remove some invasives and, you know, clean it up. Yeah, it's great.
One more question, if anyone has it.
Yeah.
Seagrass. You mentioned invasive seagrass.
Yeah.
Is it more resilient to pH levels, global warming, silt pollution? And does it perform the same function as native seagrass?
I'm gonna go to it real quickly. It's in the same genus. I don't know the answers to your questions. This is relatively new and it's being monitored actively to see if this is like a real problem. I don't know how much it has spread in the last year.
It's spreading throughout the state. You can find it right up.
Oh. No.
Well, there's been so much seagrass stuff in this big bay. There's a wide open niche.
Yeah.
If it's more durable and tolerant of lower light conditions, it's gonna have an advantage.
It's either gonna be Australia or the Indian Ocean or something, probably.
Sea turtle. Yeah, turtles over there.
That's a good question.
All right.
I don't know.
So thanks again, Melissa. Thanks for back from James. This also was that native plantain, right?
Yeah.
So we still have tons of food. Grab a sandwich, buy raffle tickets. We'll start the raffle in 5 minutes. I'm sure Melissa can hang out for 10 minutes.
Yeah.
Shut it down.
I was gonna take this with me. Yeah.
Don't ever say algae again.
We need you for other
I'm
things.
a trained phycologist and this.
I'm a Miamian. We got an S to everything.
I know.
You good with that?
Algae is.
You want to help me bring it out?
I'll-
Black, or just leave it
so
out?
hard.
Let me take it apart.
It's so hard.
I know.
Thanks.